In this lesson, we will be investigating the uses and applications of biotechnology. Many of these applications relate to GMOs, or genetically modified organisms. Studying DNA in general has opened many doors. Watson and Crick were the ones who detailed the structure of DNA back in the 50s. Since their discoveries, we have made leaps and bounds forwards. We may not yet be the Jetsons, but many of our technologies today are out of this world. It has been claimed that we live in the age of the gene. Articles about DNA and biotechnologies are in most every news source you find throughout the media. It is important to note that humans have been modifying the genetics and the genomes of organisms for a very long time. With the advent of agriculture, we see old school types of manipulations being used. Selective breeding to produce traits that we desire has long been used. One good example of this is if we look at corn. Wild corn, the original, was very small. The kernels were easily broken off of the stalk so that they could disperse quickly. We as humans have selected for the monster corns that we see today in the grocery stores. These kernels are much larger, have a much larger starch content to them, and there are hundreds, if not thousands, of corn kernels on each stalk. And those kernels generally stay put until they get very dried out. They don't shatter and explode like wild corn does. Humans are notorious for choosing traits that suit our needs. Another example is the evolution of dogs. Dogs evolved from a wolf-like ancestor. We have bred dogs based on certain traits to give us many different breeds, all the way from Chihuahuas to Great Danes. The biotechnologies we have been discussing are just new tools used to make modifications to an organism's genome. This is something that, again, humans have been doing for a very long time. We are going to be looking at biotech applications in three major fields: agriculture, medicine, and forensics. One limitation of selective breeding is the time frames it takes for these modifications to be made. Each modification must go through generations of trial and error to get the desired traits to be as strong as we would like them. Modern biotechnologies can rapidly increase and change these traits with just a few generations involved. In agriculture, some of the major benefits that we see are increased yields, crops that are more eco-friendly, and crops that are more efficient in their ecosystem. Your book poses this question: how might a genetically modified plant help 500 million malnourished people? The answer has to do with vitamin A deficiencies. Without vitamin A, we see blindness, first in night vision, and then complete blindness if the deficiency is too extreme. Beta-carotene is a compound that turns plants and other things yellow. This nutrient can be transformed in our bodies into vitamin A. Researchers have been able to add the gene for beta-carotene into white rice, which has increased its vitamin A content almost 25-fold. The donor organisms for this gene were daffodils and bacteria. Because of the beta-carotene, this new rice has a golden or yellowish color to it. Recent studies have shown that this rice is not as productive as other strains of genetically modified rice. The yields are not as high. However, research in the Philippines have shown that golden rice has decreased some cases of vitamin A deficiency. This plant, a modern GMO, still has some hope of possibly helping millions of people around the world. GMOs are all around us in the United States. We consume many products that have been grown from GMO plants. For example, if we look at corn, 86% of corn grown in the U.S. is genetically modified. 93% of cotton and 93% of soybeans are also modified. While some of these plants have been modified to increase yields, many more of them have been modified to either be pest resistant or herbicide resistant. These modifications help reduce the amount of pesticides used in the fields. It can also help reduce things like costs overall of producing food and help decrease the loss of topsoil to erosion. One of the most common forms of insect resistance that we see in these GMO crops is the production of Bt crystals by the plant. The gene for Bt crystals was originally found in bacteria. When consumed, these Bt crystals kill off the insects. The gene for the Bt crystals has been inserted into plant genomes. So now these plants, such as corn, potatoes, soybeans, will produce the Bt crystal, which makes them resistant to damage by insects. This type of resistance helps increase yields because there is less of the crop lost to the insects. Because of this increased yield, we now see more food and more products for people to use. Some of the downsides of this type of GMO is resistance. Some insects evolve resistance to the Bt crystals, just as bacteria evolve resistance to antibiotics. However, many measures have been put into place to counter this resistance. One way is to make sure that the plants are producing enough Bt crystals, a high enough dosage to kill all of the pests. Another is to plant non-GMO areas in the fields where these pests can have a refuge away from the Bt crystals. This keeps variability in the population so that resistance does not evolve, or at least not as quickly. Herbicide resistance is another example of a GMO modification that we are seeing very commonly in agriculture. One example is Roundup Ready crops. These crops have a gene that makes them resistant to the herbicide Roundup. Roundup kills weeds and many plants in general. With the Roundup resistance gene, these crops are now able to be sprayed with Roundup and they will not be harmed by the poison. All the other plants in the field are not resistant or are susceptible to Roundup, so they will be killed off. This helps increase yields because our crops no longer have to compete with the weeds. One of the major downsides to this herbicide resistance gene is that weeds are developing resistance as well. This is a larger issue than it was in the insects, and there are a few strategies to overcome this problem. We are seeing more and more GMOs coming into play every year. This year, we will see the sales of non-browning GMO apples. These apples are scheduled to hit some grocery store shelves in February of 2017. These apples were produced using a new technology, the CRISPR-Cas9 system. Plants are not the only organisms that are being modified and becoming GMOs. Transgenic salmon have been developed. These fish grow faster than their non-GMO counterparts and grow larger to produce more meat. These salmon carry a version of the growth hormone gene that functions year-round rather than primarily in the summer. This gene was isolated from a cold water fish species and injected into the egg of a salmon. The superfish can be raised more quickly and uses significantly less food than normal salmon, reaching market size within 18 months rather than 24 to 30 months. In taste tests, consumers cannot tell the difference between the transgenic and non-transgenic salmon. The benefits here are a larger fish that is producing more food for people. Some of the concerns lay in the ecological implications. If these GMO fish were to escape from the farms, they would breed with natural populations, or this could cause problems for their species and other species. These larger fish may outcompete other groups by consuming more food, and they may be too big for normal predators to catch them. It is unclear what the outcomes would be if this fish escaped from the farms. However, these salmon were approved by the FDA for sale back in 2015. So, we have seen both benefits and risks of each type of GMO. Newer research is showing that there are no food safety issues. Several studies have been done that show there is no harm in eating GMOs. Other implications and fears may be more justified, such as the ecological concerns, pest resistance, and contamination of natural populations with the modified genes. It's important to note, though, that GMOs produced with new biotechnologies are not the only ones that have had problems. This featherless chicken was bred using artificial selection. The bird was bred to be cheaper for farmers to raise and therefore cheaper for consumers to buy. Because of the lack of feathers, this bird does not produce as many dusts or mites that cause respiratory issues in caretakers either. However, there were unintended consequences for this poor bird. Not only do the fellow barnyard fowl laugh at this poor guy, but these birds are also vulnerable to mosquito attacks, to parasites, to diseases, and they're ultra sensitive to the sunlight, easily getting sunburns. These guys also have difficulty mating since the males are unable to flap their wings. Researchers are currently working to address these problems. A second field that GMOs have become prominent in is in medicine. GMOs can often be used to make life-saving drugs or products. Diabetes is a common disease in the United States. In diabetes, the body cannot produce insulin, a hormone that helps break down and store blood sugar. Complications from diabetes can include vascular disease, kidney damage, and nerve damage. Until the 1980s, insulin was actually harvested from livestock sent to slaughter. This chemical is produced by the pancreas of the body. So the pancreas of cows and pigs had to be harvested, the insulin collected, and then purified or isolated before it could be packaged and sold for diabetics. In 1982, a transgenic E. coli bacteria was produced. This bacteria was able to produce human insulin. They inserted the gene for human insulin into these bacteria so that they could produce insulin quickly, easily, and efficiently. Now insulin is produced in huge quantities. This process has opened a door for many other GMOs to be produced. Bacteria were some of our first transgenic organisms, but it has expanded further. Another interesting case is the production of human growth hormone, or HGH. This hormone was previously harvested from the pituitary glands of cadavers. HGH stimulates protein synthesis, increases utilization of body fat for energy to fuel metabolism, and stimulates the growth of virtually every part of the body. Insufficient growth hormone can be problematic. Usually this is due to pituitary malfunctions and can lead to conditions such as dwarfism. HGH is now produced using transgenic bacteria, similar to the insulin case. We can now produce HGH in large quantities, which makes it available to more people who need it. HGH is used to help treat dwarfism and certain conditions in AIDS patients. It has also been seen in sports scandals. This hormone, because it is produced naturally, is somewhat difficult to detect, so some sports athletes use it illegally to help bulk up. Gene therapy is another interesting case where information about GMOs has been very useful. Gene therapy is the process of inserting functional genes into human genomes to help replace the dysfunctional gene that was there previously. So this process has had more notable success in diagnosing and preventing diseases than it has in actual curing diseases. There's only one case that I've seen where it has been able to cure a disease. Generally, gene therapy and information from it are used in genetic screening of fetuses, newborns, and adults. This can help us with three big questions. The first question that gene therapy can approach is, are a given set of parents likely to produce a baby with a genetic disease? Many genetic diseases are inherited when each parent carries an allele for that particular disease. Things like Tay-Sachs, cystic fibrosis, and sickle cell anemia are all recessive disorders, which means both parents have to have at least one copy or one allele for that disease for it to show up. Genetic screening and genetic counseling can help with testing of embryos to figure out if a baby has a good chance of having one of these diseases. A good example of success in this field is the genetic screening, counseling, and testing of embryos for Tay-Sachs disease. This has decreased the occurrence of Tay-Sachs by 75%. Tay-Sachs disease is a deadly disease that affects young babies. Tay-Sachs disease can lead to progressive blindness, decrease in muscle strength, paralysis of muscle function, seizures, muscle stiffness, and eventually it will lead to death, usually no later than the age of 4 years old. Another question that can be addressed is, will a baby be born with a genetic disease? So we can directly screen embryos for diseases such as cystic fibrosis, sickle cell anemia, Down syndrome, and many others. This process is usually an optional situation. When a woman is pregnant in the first trimester, a doctor will offer to do genetic screening. A simple test run can help screen and determine if the baby has some of these diseases. Now, these screenings are not 100%. Sometimes you can tell, sometimes you can't, and the disease may be present even if the test says no. But it does give us a better idea of some of the genetic problems that a baby may be born with. A third question addressed is an individual likely to develop a genetic disease later in life. There are genetic screening processes for things like breast cancer, prostate cancer, and skin cancer. In this process, DNA technologies are often used to help detect disease-causing genes or mutations within those genes. For example, we mentioned the BRCA1 gene. Mutations in that gene can cause an increased risk in breast cancer. Screening for these diseases can help with early detection, which may be life-saving if cancer does hit. However, just because you have the likelihood of developing a disease such as breast cancer does not mean that you have a guarantee. Gene therapy has led to many ethical dilemmas. There's a question about discrimination. Once doctors or insurance companies know you have the likelihood of developing a disease, will they discriminate against you, such as some issues we used to have with pre-existing conditions? There's a big question about how should we proceed with this information. We've mentioned that gene therapy has good success for preventing and detecting diseases. However, it's important to look at why gene therapy has such a poor record of success in curing diseases. There are 4 major reasons that gene therapy has difficulties. 1, it's hard to get working genes into the specific cell where it is needed. This process is very complicated, and often we wind up with genes going in the wrong direction. Second reason is it's hard to get a gene into enough cells to make a difference. If only a few cells receive the functional gene, you may not make enough product to actually make a difference in your body. The third reason is it's hard to target specific cells. If we cannot target the cells we want, we may wind up with cells in random places of our body producing things like insulin that they don't need. The fourth reason, that it is difficult to regulate gene expression. We've seen that gene expression, process of transcription and translation, can be very complex in eukaryotic cells. If a cell does not have the gene placed exactly in the right spot, we may have trouble getting that new gene to turn on or turn off. This can cause other problems. Another interesting idea that has come from DNA technologies is looking at the process of cloning. So, are there any medical justifications for cloning? Many would argue yes, particularly in the process of cloning organs. This would reduce problems with long transplant lists, often saving lives if we have enough organs for those people. There's also the possibility of custom-made organs, ones that are designed from tissue or cells of your own body. This would mean no rejections. The other question is, what about cloning whole organisms? This again brings up a lot of ethical issues. We have seen cloned organisms already. Dolly the sheep is a famous example. To produce Dolly the sheep, scientists were able to isolate an egg cell from one sheep. They isolated a mammary cell from another. They removed the nucleus from the egg cell and fused the mammary cell, which included a nucleus with DNA in it, into the cell. Now that they had one cell again, they initiated cell division. The cell increased in number until they had an embryo, which was then transplanted into a surrogate mother. When the baby sheep was born, it was a clone. We have also seen other cloned animals, such as cows and cats. This is not a common process, it is very difficult, and it took many tries to get that first Dolly sheep to be born successfully. But cloning is a possibility. We also see an argument raging about cloning in other parts of biology. One example is that we have found mummified remains of mammoths. Some DNA samples have been removed from these mammoths, and some scientists have suggested the idea of cloning these extinct animals. This again brings up a lot of ethical issues. Do scientists have the right to clone dead organisms? Or do we have the responsibility to clone organisms that we as a species have caused to go extinct? Another argument is in favor of cloning things like dodo birds, a species that humans have driven to extinction. Many scientists bring up the idea of a Jurassic Park. If we start cloning many extinct animals, will we just create bigger problems for ourselves? Will we disrupt ecosystems by introducing things that are not there anymore? There are arguments on both sides of this issue, for cloning extinct things and against it. The last field we'll look at for DNA technology and GMOs is the field of forensics. DNA is an individual identifier. DNA is being used more and more in forensics to help us solve crimes, identify victims, and to even exonerate those who were falsely accused. One of the first cases where DNA evidence was used to convict a criminal was back in 1987. Colin Pitchfork murdered and raped two young 15-year-old girls. He nearly got away with his crimes because someone else confessed to the murder. However, DNA evidence collected from the victims showed that he was actually the killer. Police and scientists were able to collect DNA samples from many suspects and compare those DNA samples to the DNA found on the victims. Colin was a perfect match. Because of this evidence, he was put away for his crimes. In forensics, DNA evidence is often in the form of a DNA fingerprint. We can use this to identify suspects, exonerate innocents, ID victims in situations where bodies are unrecognizable, and even detect pollutants such as bacteria or other organisms in waterways or soil. To create a DNA fingerprint, we have to figure out regions that are unique in each DNA strand. 99.9% of DNA is exactly the same between you and your neighbor and every other human. Yet we do have certain unique strains. One region that has much variability and some uniqueness to it are STRs, short tandem repeats. These regions have different alleles, and the allele is how long the short tandem repeats are. You get two alleles, one passed down from mom, one passed down from dad. So your combination of alleles will be unique or different from other people around you. To make a DNA fingerprint, we have to use our DNA tools. So we start by chopping out STRs using restriction enzymes. We amplify the small amount of DNA that we have using PCR, and then we separate the DNA strands using a gel electrophoresis technique. The separated pieces are at different lengths and will travel different distances in the gel. We can then compare these DNA fragments and DNA gels to other people to determine if you were at the scene of the crime. DNA fingerprints, again, are distinct to each person, but they can also tell us about your relatives. We can use these kinds of information to determine things like paternity as well. This kind of forensic work has become commonplace. We see it in crime shows all the time. We can also look at places like the UK. They are one of the first to set up a database containing these DNA fingerprints. They have genetic profiles for more than 2 million people. This is another area that brings into the question ethics. This type of database has been able to decrease the time it takes to solve crimes and is particularly useful for repeat offenders. It can also be used for helping find lost children or to identify children when they go missing. However, many people don't like the idea of the government having this kind of record of them. This is an example of a DNA fingerprint that is more complex than the average one that's used. This is the kind of fingerprint that the UK is keeping on database. So they are not actually keeping a DNA sample from each person, just this picture of what the DNA strands look like. So, in summary, this lecture has covered biotechnology applications. We have looked at the fields of agriculture, medicine, and forensics.